This is Chapter 6, Part 5. Hopefully this will be the last installment of Chapter 6. We're going to talk about partnerships first. Symbiosis, it's a general term to denote a situation in which two organisms live together in a close partnership. There's three main types that we'll talk about: mutualism, commensalism, and parasitism. Mutualism is when organisms live in an obligatory but mutually beneficial relationship. Commensalism, partner called the commensal receives benefits while its partner is neither harmed nor benefited. And parasitism is a relationship in which the host organism provides the parasitic microbe with nutrients and a habitat, and the host suffers from the relationship. So an example of mutualism is in our gut, we have microbes, and some of those microbes provide, they make vitamin K. So some of those microbes make vitamin K. And we, as humans, need vitamin K in the act of blood clotting. So if you're on long-term antibiotics, you could develop some bleeding issues, theoretically. And I know that this happened with somebody I know, where they were on long-term prophylactic antibiotic therapy because of an organ transplant, and in the process, and it was years later this happened. They their hemoglobin went way down, and they couldn't figure out what was going on. They did, all these tests, imaging to see if there was any bleeding internally, and they couldn't figure out what was going on. And as it turned out, they were somehow losing blood, and I can't remember the process, but they were losing blood, and it was due to the fact that these microbes were wiped out from their normal flora, which made vitamin K, were wiped out, and the person was no longer making vitamin K, so they I don't even know if they checked their vitamin K levels. I mean, there was a series of things that went on. They were trying to figure out if the wife was poisoning this person with rat poison, because that'll make you bleed. Anyway, long story short, they determined by accident that this person, once they gave them vitamin K, they were better. They stopped losing blood, and the issue was resolved once they went off the antibiotic. So it's kind of interesting the way things happen sometimes in the medical field, because there was a room full of doctors at Johns Hopkins that couldn't figure this out, and it was kind of an accident the way they found out. But anyway, so know each of these terms as to be able to define them. Antagonism, if you antagonize your sibling, okay, so it's a give and take. It's kind of like a competition, right? So an association between free-living species that arise when members of a community compete. Antibiosis, production of inhibitory compounds such as antibiotics into the surrounding environment that inhibit or destroy other microbes in that same habitat. So there are microbes that are able to do that. And the first microbe, through this process, has a competitive advantage by increasing the space and nutrients available to it by doing this. This is just an example of antagonism, this here. Synergism, so everybody's working together. It's an interrelationship between two organisms that benefit them, but is not necessary for survival. So it just kind of makes them stronger together. They're fine separately, but they're better together. Together, the participants cooperate to produce a result that neither could do alone. Examples are gum disease, dental caries, which are cavities, and some bloodstream infections that involve mixed infections. And basically, the main example of this is biofilm formation. And I know that this picture is small, but it's bigger in your textbook. But biofilms are basically armies of microbes that work together, and individually, each of these pink microbes may not be nearly as damaging as when they work together with these purple microbes, right? So they work as an army. They're much stronger. It's like, putting the Army and the Navy and the Marines and the Air Force all together, and the whole process, the whole group is stronger than if it was just one individually. So biofilms are the epitome of synergy, mixed communities of bacteria and other microbes that are attached to the surface and each other. During the formation of a biofilm, the pioneer colonizer attaches to a surface, and they're able to send out signals to other microbes that then attach to those bacteria or, polymeric sugar or protein substances secreted by the microbial colonizers. The attached cells are stimulated to release chemicals, or like I said, signals, as the cell population grows, and that attracts even more. So a biofilm is really, really hard to treat because you have multiple, types of microbes there that are like, it becomes a super infection almost. If it's not really the right term to use because that's used for other things, but it is much harder to treat a biofilm than it is to treat an individual infection from a specific microbe, a single microbe. So bacteria in a biofilm behave and respond differently than free-living bacteria. And that's because different genes are activated. So the biofilm, like I said, is a much stronger infection. So the study, I just flipped from here to here. I haven't skipped anything big. So the study of bacterial growth. So we're going to talk about how bacteria grow. And I want to clarify that I talk about growing. When we think about growing, we think of going from small to big, and that's not necessarily what we're talking about here. When bacteria reproduce, we'll say, it's called binary fission. And that is when one cell becomes two. So basically, the parent cell enlarges a little bit, its chromosomes duplicate, and then it starts to pull its cell envelope together to the center of the cell, and the cell wall eventually forms a complete septum, and then the cell becomes two. That is the summary of binary fission. So when bacteria multiply, it is referred to as binary fission. Okay, binary multiply or reproduce or grow, but again, growing not meaning a little kid getting big into an adult. Okay, so here are the steps illustrated of a rod-shaped bacterium. So you have a young cell, it's doing its normal thing. It gets a little bigger, and the chromosomes duplicate. Okay, so now they're exact copies of chromosomes. Then this little septum starts to form. The protein band forms. I call it a septum, but you could always combine these two steps here. So that septum formation happens, and then eventually the cell breaks apart, and then you have two separate cells. These are the daughter cells. This is the parent cell, and these guys are exact copies of each other and of this. Okay? So the generation time or doubling time is the time required for a complete cycle of binary fission from parent cell to the two daughter cells. The generation is when we increase the population by a factor of 2, so from parent to daughters. And as long as the environment remains favorable, the doubling effect can continue at a constant rate. So generation time can vary depending on the organism. 30 to 60 minutes is the average generation time. Shortest is 10 to 12. Mycobacterium leprae has a generation time of 10 to 30 days, so that generation time is very long. And environmental bacteria have generation times measured in months. Most pathogens, ones that make us sick, have generally short generation times. Growth curve is a predictable pattern of bacterial population growth in a closed system. So it can be measured by, basically we put cells in a sterile broth, we incubate it over a period of several hours, and then we sample the broth at regular intervals during that incubation. So like at 30, 60, 90, 120, and so on. And then those samples that we take at those regular intervals are then plated or inoculated onto a solid medium, and then we count the number of colonies present on each auger after we incubate those. And this is just an example of showing how that happens. Initially, nothing's going to grow initially, even though we've inoculated this, right? So let me just verify that this is, yep. So this is inoculated, and this has bacteria in it, and there's no growth initially, and you're like, well, why wouldn't there be growth? Because we put the bacteria in there, and you're putting it on a plate and you're incubating it. And that's because if we sample it right away, then it takes time for those bacteria to kind of get acclimated to their environment. So and then you can see at each interval after how the growth increases. So stages in a normal growth curve, it starts with the lag phase, and that is the flat period of growth. Due to what I just said, period of adjustment, cells are not multiplying yet. So you're going to have to come up with these and know what happens in each of these phases in the stages of the normal growth curve. The next stage is the exponential growth, or the logarithmic or log phase. That's when the growth curve increases geometrically, and it will continue as long as cells have adequate nutrients and the environment is favorable. The next stage is called the stationary growth phase, and that is when cell birth and cell death rates are equal. Cell division is starting to slow down, and that is because there's a depletion in nutrients as well as oxygen, plus an excretion of organic acids and biochemical pollutants in the growth medium. And think of it as if you've waited a while to clean out your fish tank. It starts to kind of get yucked up, right? That's the same idea. Eventually that fish will die because it's swimming around in its own waste. That's the same idea with bacteria. Eventually its own wastes will smother it out, will kill it. So to start from the first part again, we have lag phase. It's the flat period of growth. Nothing's happening a whole lot because the cells are getting adjusted to their environment. Then you have the exponential growth phase, lots and lots of growth. When I say growth, remember, it's basically they are going through that binary fission. Not growth as in the cells are getting bigger necessarily, it's that they're multiplying. And then we have the stationary growth phase where it's slowing down. Birth and death are equal. And then the last phase is the death phase. Cells begin to die at an exponential rate due to the buildup of wastes. Speed at which the death occurs depends on the resistance of the species and the toxic conditions. Basically, viable non-culturable state is many cells in the culture in the death phase stay alive but are dormant. So we have to decide whether they are viable to count, but if they're not multiplying, then they're not still growing. So, that's kind of confusing idea, but just know that at that point they're dormant. So here this is laid out on this diagram. So you say your lag phase, and then lots and lots and lots and lots of growth in the exponential growth phase, and then we're leveled out on the stationary phase, and then during the death phase, more cells are dying than growing, and there are a few viable cells left. All right, so I'm going to try to get through this in like 50 seconds. So the tendency of populations to exhibit rapid or phases of rapid growth, slow growth, and death has important implications in controlling microbes in the environment. Microbes in the exponential growth phase are more vulnerable to antimicrobial agents and heat. I feel like that's a question that comes up in one of the assessments. Cells in the growth phase are more vulnerable to conditions that disrupt cell metabolism and binary fission. I also feel like that's a test question. So growing cells are more vulnerable to growth inhibition and destruction. And you know what? I meant to take this out. So you can just review these last 3 slides and not be worried too much about these. And that's it. That's it for chapter 6.